So we're starting the recording for Glycolysis and Cellular Respiration, Chapter 8. You know that we're stopping the test after we finish discussing glycolysis. So how do cells get energy? Most cellular energy is stored as ATP. You should remember that ATP is called the energy currency molecule for the cell. That means ATP is used whenever cellular work is needed. Remember, work equals energy. You have to have energy to do any type of cellular work. Cells use glucose to make ATP. You should know what glucose is just by looking at the word. If a word ends in os, OSC, that means it is a sugar. So we're going to talk through glycolysis, the process, as to how cells break down glucose through glycolysis to make energy. There's two stages of breaking down glucose. We're going to stop the discussion with glycolysis for your test. So glycolysis and cellular respiration are the two breaking down glucose. Glucose is an energy storage molecule. That means that glucose in itself, once broken down, produces ATP. The glucose stores the energy. Your body does not start breaking down glucose until you need that energy. All cells use glucose as their energy source, whether it's plant, animal, bacterial, cell. And glucose can be stored if you have too much glucose, too many carbohydrates. It's stored as either glycogen or fat. Fat is a long-term energy storage for the body. So the written down overview of glucose breakdown, I'm going to just talk about bullet 1 for this test, glycolysis. It begins with the breakdown of glucose. When glucose is broken down, one of the end products are two molecules of pyruvate. A little bit of ATP is made during the process of glycolysis, but not a lot of ATP. Key note for glycolysis is it does not require oxygen. That's called anaerobic. When a process does not require oxygen, it's called an anaerobic process. As well as you should note that glycolysis occurs in the cytoplasm of the cell. So whether or not oxygen is available depends on what happens after glycolysis, but again, that's going to be in your next test, so I will wait and discuss that at a later date. So what happens during glycolysis? I'm going to read through this, but then I'm going to look on page 131 in your textbook after I read through, and you know that's what you need to know for your test. 131, memorize it, understand it. If you have any questions, you should email me or send me a text message. So the first thing, glycolysis has two parts. The glucose activation. During glucose activation, that means that ATP has to be used to activate the glucose molecule. And then energy harvesting. That means that with the energy harvesting phase, you will actually end up making some ATP. So the first phase, you use ATP. The second phase, you make ATP. Before glucose can be broken down, it has to be activated. Remember, glucose on its own is not a reactive molecule. It becomes reactive when you add phosphate to the glucose. The only way you can add the phosphate is to get them from ATP. Glucose is converted to fructose bisphosphate. That fructose bisphosphate is very unstable. That means it's highly reactive. The fructose bisphosphate can be split into two molecules of glyceraldehyde 3-phosphate, which we call G3P in class. And then the G3P undergoes a series of reactions to convert it to finally pyruvate. So two ATPs are made for each pyruvate for a total of 4 ATPs. And it says in your notes here, 4 ATPs are made in the process of glycolysis, but you have to remember that we use 2 at the very beginning of the process. So you use 2, you make 4. 4 minus 2 is 2, so you really only gain 2 ATPs for the cell. You also are going to gain 2 NADHs, which you know NADH are energy carriers, and you're going to see with the next material in the chapter that NADH goes to the Krebs cycle. Now, page 131. First, let me say you see the YouTube video here that we listened to in class with the Glycolysis rap song. So look through that, and maybe that will help you. But I'm going to continue talking from this slide on page 131. So you'll remember in class that we acted out glycolysis. We had 6 people lined up. Each of those people represented a carbon because glucose is a 6-carbon sugar. Its chemical formula is C6H12O6. But what you see in your textbook are just the circles that represent the molecules. You'll remember that glucose by itself is not reactive at all. It has to get to be energized and become reactive. So glucose enters into the cell, and to make it reactive, to be able to get the energy that we need from it, ATP is added. So for a chemical formula, a very simple formula, you could write glucose plus ATP yields glucose 6-phosphate plus ADP. So in that first step, ATP entered in, gave off a phosphate. The phosphate was attached to the number 6 carbon of the glucose, so it became glucose 6-phosphate. Now, the glucose is going to flip onto a mirror image of itself, and it's going to become fructose 6-phosphate. So it's just being rearranged in that basic step. The next more important step is another ATP comes in. It donates its phosphate to fructose 6-phosphate, making that molecule now fructose 1,6-phosphate. Remember, the 1 and the 6 just refer to the number of carbon where the phosphate is attached. Now at this point, glucose is a very unstable, fructose is a very unstable molecule. So it wants to react. Glycolysis has not happened yet because glycolysis literally means splitting glucose, and we haven't split glucose yet. All we've done to glucose is change it around a little bit by adding on some phosphates and rearranging it to make it into a reactive molecule. We have invested at this point 2 ATPs in this process. Now the next step is glycolysis. The sugar glucose is split, and it becomes G3P and DHAP. So DHAP cannot go any further in this process, so automatically DHAP is converted to G3P. So what we have now are 2 G3P molecules, glyceraldehyde 3-phosphate. You'll see that in your textbook. You have your 2 G3Ps. Now for the next reaction, those 2 G3Ps are going to react with 2 phosphates, as well as react with 2 NADs. The NADs are going to pick up some electrons, hydrogen, and NAD is going to become NADH. When it becomes NADH, the NADH is going to go away from the process and enter into the Krebs cycle as an energy carrier. The phosphates are going to come in and react with the G3P and convert that into 1,3-bisphosphoglycerate. So you see that step in your text. The next step is 2 more ADPs come in and pick up a phosphate. When ADP picks up a phosphate, it becomes ATP. So ATP is an energy carrier molecule as well. After that happens, you have one phosphate that's now been taken away from the molecule, and you have phosphoglycerate. Phosphoglycerate is converted, rearranged. You see in step number 8, after another rearrangement, that's what you see in between phosphoglycerate and phosphoenolpyruvate. The phosphates have been rearranged, so you just moved your phosphates there to the second carbon instead of the third carbon. So phosphoenolpyruvate is then converted to pyruvate, or in some textbooks you'll see it called pyruvic acid. When ADP comes in, picks up the phosphate, and converts that to ATP. So all of glycolysis has occurred to make your energy carrier molecules. It's occurred to make ATP. It's occurred to make NADH. In this process, you can see from your text, we made a total of 4 ATPs, as well as we used 2 ATPs in the energy investment phase. So you net gained only 2 ATPs. So glycolysis really does not make much energy. But as you will learn in the next cycle, the Krebs cycle does make, along with the electron transport chain, a little bit, especially the electron transport chain, more energy than glycolysis. So you should understand backwards and forwards. Page 131, glycolysis. This page will be 50 points on your test. And you'll also have multiple choice questions and some short answer on your test about glycolysis. Again, if you have any questions, you should send me a text or send me an email and let me know.